talking about tRNA and about how tRNA has two points of attachment that are really important to us. One of those is where the amino acid will attach up here. And the other is where it will attach to the mRNA at the other end of it. We also talked about the wobble idea, that being that the first two letters of the codon, remember a codon is a three-letter sequence of nucleotides that is associated with an mRNA that will code for a particular amino acid. You have to have three letters. The first two letters are more important than the last one. The last one tends to wobble, meaning that you could have usually a couple different, in some cases you could have all four of those letters could be the last nucleotide, or it could be the last codon, and it would still give you the same outcome. So I talked about proline and how proline could be CCC, but it could also be CCU, or it could be CCA, or it could be CCG. Okay? So the last one's kind of not as important because you could substitute in any of the other letters for it. So tRNA, in order for tRNA to become, quote, charged, which means it's now activated, it has to go through a couple of steps because it has to have, in order for it to do its job, it's got to pull with it a amino acid. Well, it has to have that amino acid attached to it. And it does that in a step where there's an amino acid that's going to catalyze that reaction. It's going to put those two things together. And that amino acid is called tyrosyl tRNA synthetase. Yeah, you don't have to remember that name. You just have to remember there's an enzyme that's job is going to be to attach the amino acid to the tRNA. That's all we really care about. Okay, so here we have in the second picture, we have this was one. Now we've got two. And notice how we're using some ATP to do that. So it takes some energy to do that. We're going to covalently bond on that amino acid to the tRNA. And whenever we do that, whenever we attach a bond like that or put something on with a bond like that, it's going to require some energy. And then the last thing is it just gets released. And now I have an activated tRNA that's ready to go to the ribosome and carry with it its assigned amino acid. So now that our tRNA is charged, it's ready to rock and roll. Now we can talk about the other end of that, and we can talk about the ribosome part. So ribosomes facilitate the coupling. In other words, they help the tRNA anticodons. Remember, they're anticodons that are lining up with the codons of the mRNA, but it helps it line up so that happens correctly. There's two parts to a ribosome: a large part and a small part. They're both made of protein and ribosomal RNA. We call that rRNA. Okay, so they are nucleic acid and they're protein. Bacteria and, I don't know why I keep saying bacterial, prokaryotic and eukaryotic ribosomes are somewhat similar in terms of their ribosomes, but they are not identical. Okay, so that is important because those differences are significant in that the way many of our antibiotics work is to target the ribosomes and do something to keep those ribosomes from doing their job. Because if the ribosomes break down, if that machinery breaks down, that means that the proteins can't be made to make new bacteria. So the infection won't spread. So what antibiotics are doing is it's keeping the bacteria from multiplying. Because if you don't have more raw materials, you can't make more bacteria. And so You won't get rid of the bacteria that's in your body, but you'll keep it from spreading. And eventually that bacteria that's in your body will be destroyed by our immune system. So that's an important feature of antibiotics. They work on prokaryotes, but they don't work on us. Okay, the ribosome has 3 binding sites. It has a P and an A and an E site. And I'm just going to show you a picture. I'm going to go a couple slides forward and show you that first. And then we'll come back and we'll talk a little bit more about this. So here's my ribosome in brown. I think of it as a hamburger bun because I love food and everything I do is a food analogy. So we've got a top half here. That's our large subunit, right? It's large. That's the top half of the bun. And then you got the other skinny one underneath it that is going to be our small subunit. Okay. RNA is going to be kind of the lettuce or cheese or whatever that goes through here. The mRNA is going to be going through here. Oh, boy, I can't draw a straight line to save my life. And within that large subunit, there are these 3 binding sites. There's an E site, a P site, and an A site. And they all do something a little bit different, but they're all working with tRNA. Their job is going to be to help guide that tRNA so it's in the right spot and so it doesn't falter and it moves to the right location at the right time. And let me see which slide I want to go to next. Okay, let's do that. Maybe, or should I do that one? Yeah, no, this one's better. Okay, so here is the ribosome now, and now we have an mRNA in here down at the bottom. This little guy here. And notice that we have a tRNA that's slotted up in this P site. The P site and the A site are probably going to be, well, they're all important, but the P site and A site are going to probably be the most important because what happens is they will enter, each tRNA will enter in the A site. And after they enter in the A site, they're going to move over. They're going to shift one site to the P. And after they do that, another A will come along and another tRNA will come and it will bind in the A site. Okay. When that happens, the 2 amino acids that they have been carrying will detach from where they are attached to the tRNA and they will attach to each other. So here there's a little like leash that's holding the tRNA or the tRNA to the amino acid, and that's going to release and another bond will form. Do that in green here. That's between those 2 amino acids. Okay. And that's going to keep happening that this guy here is going to shift over one to the P site. The P site one is going to shift over to the E site. And after that happens, it leaves. What do you notice that's different about this one versus, I'm going to clear the screen here, this one versus this guy here? What's the difference between those? What is attached to one that's not attached to another? Amino acid. So there's an amino acid attached to the one on the right here. It's charged. It's ready to go. And then here, it's released it. And now it's ready to go back, find its correct amino acid based on its code, get another one of whatever one it carries. It's always going to carry the same thing. And then it'll become charged again and it can come back to the ribosome and repeat that process. And I know I'm getting a lot of like, oh my God, I have no idea what she just said. That's okay, because that's part of the process is learning all this. We're not there yet. There's 3 stages to translation: initiation, elongation, and termination, just like there was in transcription. Each of the stages requires some type of factor. It's usually a protein that is going to help with the translation process. And also key in this is that there's going to need to be some energy as well. Sorry, I was trying to do that. Transcription and translation are very energy heavy process. Gene expression is a very energy heavy process. So we're going to talk at some point about gene regulation in eukaryotes. And there's a lot of ways that you can regulate whether or not this process occurs, and you can shut it down at multiple places. Because if there's something going on and you no longer need a particular protein to be made, you want to shut it down immediately because you don't want to waste that energy. Okay? And that's really important in any living system, but especially in living systems that are worried about day to day. Are they going to get enough energy? Organisms that are out in the wild that need to get energy from a day to day basis. If they don't have enough, they may starve and then they don't reproduce, and the species can suffer because of that. So making sure you shut this down in a timely fashion is going to be important. We're going to talk more about that next week. All right, so come on, there we go. Always, the first codon is AUG, A-U-G. It codes for a special kind of modified amino acid that is MET. MET stands for methionine. You don't need to remember the methionine. Well, I guess it's not a bad idea to remember the methionine. You might actually see that sometime. Methionine. Okay. So that AUG will code for the amino acid methionine, and that's always, because that's its job. That's just its job, is to be the start. You always know if you see that code that must be the beginning of the protein. The first thing that's going to happen is our little hamburger bun, the small piece, is going to attach to the mRNA. And that is shown in this next picture here. So here I have my lonely little first tRNA carrying its MET or methionine. And you can see that down here, the bottom one, that's my mRNA. And that mRNA is coding for AUG, or I shouldn't say coding for, it is carrying the codon AUG, A-U-G. And that will match up with UAC and MET. Okay. Now, the next thing is going to happen. So now I just have the bottom of my hamburger bun. I have the bottom small ribosomal unit. The next thing I need to do is I need to put the other piece over the top of it. I need to put that large subunit over the top of it. And that comes in and it slots in so that the methionine and the tRNA attached to it are in the P site. Okay, so the very first place that the tRNA and the mRNA is going to attach to is going to be in that P site. Okay. Where do you suppose the next one's going to come? Next tRNA is going to come. Yeah, it's going to come in the A. Yo, that's cray-cray. It's going to come in here. I can't make my sock puppet very well. You kind of get the idea. Kim. She's got to kick her things. Yeah. So that blue thing there that's very poorly drawn is my little sock puppet. And that's going to be my second tRNA carrying what? What's it carrying? What's it carrying? It's carrying an amino acid. Yeah. I'm going to make it out here so we can actually see it. And it's going to have a probably different shape, right? Because it's a different amino acid. It's a different codon. And I think that might be shown here in one of the next pictures. So after I've initiated the whole thing, now I'm going to elongate the chain. And elongate just means I need to bring more tRNAs to the ribosome. And I need to attach those tRNAs I need to attach the amino acids to the developing chain of amino acids that I have. So during elongation, amino acids are growing or added by one at a time to the growing chain. Each one involves proteins called elongation factors. We're not going to get into that piece.
Sorry to interrupt. She said she's done with all the.
Oh, okay, awesome. I was going to say that we got this beautiful recording for you, Kim, if you need to leave, but we'd rather have you here. Elongation occurs in three steps, and we're not really very interested in that. But again, just like we said before, energy, lots of energy. This process uses a lot of energy. You have to attach. Now you're taking off the bond between the tRNA and the amino acid, and you're shifting it to have another bond between the amino acid and the amino acid that's next to it. Okay, so that takes energy. Okay, just like always, translation is going to proceed in the 5 to 3' direction. Yeah, Harper.
So the amino acid chain, where is that attached to? Like, is it like on the tRNA?
It's going to be. Let me see if I can show you a picture here. So it's going to be attached to that last tRNA that you have, and then it stays attached to that. It just keeps getting transferred. It's like, no, you take it. And so the next one moves over and it takes it and passes it on. And then eventually what will happen is the whole thing will break apart, and the amino acid sequence will become a polypeptide, and that will break off from the last remaining tRNA that's holding it together. One thing I didn't really cover very well, and I want to go back to that before I go forward again, is this idea of we have, what's the last site called? We have E. Okay. So what's going to happen after these two get attached together? And I think the video will help you with this. I'm going to show you a video. That's supposed to be a peptide bond. But after those two are attached to each other, they're going to shift. So the one that was in the P is going to move to the E. The one that was in the A is going to move to the P. Okay? And then another one will come in beside it in A. The E no longer has a amino acid, so it's going to E, exit. It gets ejected or exited, it leaves. Okay, so it's really, once it gets to that E site, it's just going to leave. Yeah.
Wait, so it doesn't have like an amino acid, so like it just like doesn't do anything?
The E, no. No, it's really just a slide. Once it slides over there, that's the cue for the tRNA to leave.
Okay, why couldn't it just like leave after P though? Or...
Well, when it's with P, it's in the process of attaching that amino acid to the chain.
Right, but like after P, like why does it slide to E? Why does it just,
I think it's because the mRNA is actually sliding over. The mRNA is sliding over, and the bun is sliding over as well in the opposite direction.
So it's E is just there.
Yeah.
So it's like it's leaving the tRNA.
Mm-hmm. It's leaving. The tRNA is leaving the ribosome, and it's going to go out. And it's going to look for another amino acid. Let me see if this. You do this one page, and then I'll show you what that looks like. The ribosome and the mRNA move along relative to each other. So you've got your bun on top of it, and the mRNA is moving like this and along there. The cycle takes less than a tenth of a second to put on an amino acid onto the chain. So in 10 seconds, or in a second, you're going to get 10 amino acids put on. So it's really quick. The whole cycle is shown in this picture here, where I'm looking at the top here first. At number 1, I've got my P site in there. And then I'm going to have another tRNA that's going to come in next to it. And it's really hard to see this because it's so small, but there's a little bond being placed. I think there's a circle. And I think that's supposed to be a diamond shape. So that's being put on in that location. So now there's a peptide bond, and this little guy down there is going to lose that amino acid. And once it loses that amino acid, the whole thing is going to shift. So the bun is going to shift down this way, and the mRNA is going to shift this way. And when it does that, that tRNA that was in the P site here, this guy here, now he's going to leave because he's done. He's done his job, and now he can move back and become charged again. Charged meaning we use some energy and we put on an amino acid with the enzyme. Okay. All right. Last step is going to be termination. Oh, there's a lot of stuff there. And in the termination, stage, it's going to be just what you think it is, which is it's going to finish. Come on. There we go. So elongation is going to continue until it gets to a stop codon. There are 3 stop codons. There's only one start codon. It's OG. There are 3 stop codons. Don't ask me what they are because I can't remember them off the top of my head. But they are in that codon chart. So if you are transcribing or translating a code and it comes to a stop, then you know you're done. You're done. The A site accepts it accepts a protein. It's not a tRNA, but it's a protein. It's called a release factor, and that release factor will trigger the whole complex: the small subunit, the large subunit, the mRNA, and the tRNAs to all dissociate from each other. And that's kind of shown in this next picture, where it's like, okay, here's my stop codon. I'm done. It's like, okay. So this release factor comes into the A site, and the whole thing breaks apart. Again, it takes energy to break it down. All these steps require quite a bit of energy. All right. Often translation is not enough to make a protein functional. We call it a polypeptide chain when it leaves the ribosome on purpose because it's not necessarily a fully functioning protein. It will start to fold on itself once it has been translated, so it'll start to fold. In other words, it will become its 3-dimensional shape that it needs to be. But that doesn't necessarily mean that it's completely done. Sometimes there are what we call post-translational modifications that have to happen to it to make it actually functional. And that is going to be dependent on what protein you're talking about. So we're just leave it as kind of generic. It's got to have some post-modification to it. Post-transcription modification, translational modification. Remember that this DNA that we had is going to code for not only the primary structure, but because of that primary structure, we're going to have a particular shape. And that will give it a particular function. Okay? Because we've got certain hydrophobic and certain hydrophilic amino acids, it means that different shapes will form out of that. And we'll actually talk about that in a specific example of how the shape of the protein changes in a detrimental way when we get to mutations, but we won't cover that until next class. 2 populations of ribosomes are found in cells. So there are ribosomes that are attached to the endoplasmic reticulum, and there are free-floating ribosomes. They are the same, but they do different functions typically. A free ribosome could become an attached ribosome attached to the ER, or one attached to the ER could vice versa go to become a free ribosome as well. But while they are in their particular location, they're going to have particular functions. So free ribosomes tend to produce proteins that are going to function in the cytosol or the cytoplasm. In other words, they're going to stay in the cell because there's no need to ship anything out. They don't need the endoplasmic reticulum. Remember, the endoplasmic reticulum and the Golgi is going to package up the protein and get it ready to ship it out to export it to go to a different location. And so that's going to be the bound ribosomes. Those bound ribosomes are going to make proteins that are going to get shipped out of the cell. And so you need to be able to form a bubble or a vesicle, a membrane around that. And so it's important for that bound ribosome to be attached to the ER so we can do that. There are also these complexes called polyribosomes, which is where you have a lot of ribosomes all working on the same strip of mRNA. Sometimes it's called a polysome. That makes the translation happen a lot faster. So, for example, if you think about it, how fast does adrenaline happen? Fast, right? Hella fast, right? It's like that. So having that ability to have multiple ribosomes producing multiple adrenaline proteins is important because if you just have a couple of adrenaline molecules, that's not going to work. It's got to be an immediate and swift response. And so that's accomplished by these polyribosomes. Here's an example of it in bacteria, I think. Yeah, in a bacterial cell. So I've got my hamburger buns. I've got a lot of hamburgers. And they're each working on the same RNA strip. It's just one's further along than the other. One just comes right behind the other. Kind of like snowplows on the interstate. You ever seen that? Where there's like a line of snowplows coming down the interstate. Maybe, maybe not. Maybe you haven't driven enough to see that yet. Bacterial cells have a very streamlined process. There's no hesitation between transcription and translation. And that means that they can make new proteins very quickly. And that means that they can make new cells very quickly. They don't have a lot to copy. They don't have a lot they don't have organelles. So they can make new proteins and they can make new cells very quickly. Remember, prokaryotes do have ribosomes. They have DNA. They don't have a nucleus. They don't have membrane-bound organelles. Okay, that's going to be one of those key pieces that's going to come up as we get closer to that AP test. Here's an example of bacterial DNA. I'll use blue for that. This is my DNA. I have these little enzymes. What transcribes the DNA into an mRNA? What makes it into mRNA? What enzyme? Polymerase, yeah. So I've got these polymerases. Here I've got several of them working. And as soon as that mRNA message is generated, it starts to get translated, and all those little dark pieces are all my little hamburger buns. So I've got lots of ribosomes very, very quickly working on a strand of mRNA. Okay, so you can produce tons of protein very, very quickly if you're a prokaryote. You can't do that if you're a eukaryote. Why not? What do you have to do? You have to edit it before it can leave the nucleus. Yeah. So it has to, remember we have exons and introns. The introns are in between. They are only found in eukaryotes. They're not found in prokaryotes. So prokaryotes can just bada boom, bada bing, slam out a whole bunch of proteins. Eukaryotes can't do that because in order to leave the nucleus to go to the ribosome, you have to cut out that extra gobbledygook. You have to cut out the introns. So that it's an extra step, takes a little bit longer to realize that process. So in eukaryotes, the nuclear envelope separates the process between transcription and translation, and there's that editing that has to happen before it can leave the nucleus. Okay. So here are some pictures of that process, and I think I still have up. These are going to be helpful when you are going to do your project tomorrow. And I'm going to stop at mutations, but I want to show the rest of this video. But it's not the one that we were watching yesterday, is it? Or is it? It is, but it's reset, so we got to find it where we were at. Oh, dude, it was not that much fun to watch the first time.
It was very helpful though. I felt very informed that.
All right, what is that? Anybody know?
That's the mRNA.
That's the mRNA. And on one side, what do I have? I have a poly A tail. I think that's this piece here. I think that's my poly A tail. And then I also have a 5 prime cap, modified guanine that's stuck on there. And this is mature because is there two colors? Are there two colors?
Yes.
In the middle.
Oh, there's one color.
Right here, right? There's only one color. There's only one color. It's red, right? There's only one color because we've already edited out at this point the introns. And I think this is where we left off, more or less. Oh, yeah, there we go. Okay, let's see if we can actually play this now. Oh, no, not with that, though. Let's see. All right, so a couple of things. What is this? What's that purple molecule there? What kind of amino acid? It's the first one. It's OG is the codon, but what's the? It's MET. It's MET. Okay. What is that? So we got some slots here. What's the slot that first one came into? That's the P site. P. P. And then what's to the right of it? Apple A. And then what's to the other side of it? E. Okay, that's my E. Probably not the best color in the world to use. EPA, Environmental Protection Agency. Hey.
Wait, is that?
Has nothing whatsoever to do with it. I used to know people who worked in EPA, Environmental Protection Agency. So that works for me. It might not work for you. Might have to try something out. Something else. All right, ready? Sure, we're ready. All right. Well, that looks much better than mine.
It's crazy. Like, my lab is doing
Yeah, it is. It looks like a. All right, what organelle is that leaving?
that. Golgi.
It's right here. It's the ER. Yeah, it's the ER. The ribosomes. Remember, we have rough ER and we have smooth ER. Rough. This is the rough ER. Where's it moving to? It's moving to the Golgi. Yep. Good. Where it's going to get processed. Oh yeah, we get that. Okay. Does that help?
Sure.
Maybe. I hope. All right.